Our speaker today is Dr. Michael Charette. He's from the Francophone Northwestern region of New Brunswick. So when you say out east, we don't mean Ontario. He's from real out east, right? Where the out east actually happens. He completed his undergrad cellular and molecular biology at Dalhousie and his PhD in biochemistry and molecular biology in the Faculty of Medicine at Dalhousie. He was then a postdoctoral fellow in the departments of therapeutic radiology and molecular biophysics and biochemistry at the Yale School of Medicine. He's been with us here at Brown University since 2014. Is that long? Wow. Where he's a tenured assistant professor of biochemistry in the Department of Chemistry and a member of the Children's Hospital Research Institute of Manitoba, CHRIM, and of the Paul Albrechtsen Research Institute at Cancer Care Manitoba, CCMB. The research he does in his lab, which I am assuming he's going to be talking about today, uses yeast models to study the function, regulation, and diseases of ribosome assembly. And I'm sure he'll do the same thing, but he's got in his bio here to thank his funders, funded by NSERC DG, Joint CFI, JELF, CHRIM, CCMB that I mentioned a moment ago, and Brandon University. So, floor is yours.
Okay, thank you very much, Dr. Malcolm. So I've given little bits and bobs of this particular talk probably previously, but right now we're at a point where this project is finishing up. It's really wrapping up. I'm also presenting it internationally next week, so you guys are my guinea pigs. The only thing is I only have 15 minutes to present it, not an hour. So there's obviously a lot more background that I'll be presenting to you today that I'm going to have to cut out. As part of the introduction kind of mentioned, one of the kind of questions I was actually asked recently by a friend of mine is, well, you know, what exactly is biochemistry and like, how does it compare to other things? And it kind of took me by surprise as a question, and I stumbled a little bit, and I kind of thought, well, okay, there's definitely an issue of size, right? So the proteins that we work on are very, very large, relatively speaking, and I have a comparison point that I'll show you once I get there in terms of scale of what I look at. But the other thing too is, and I kind of jerry-rigged this a little bit on purpose, there's a large chunk of my education that was done in medical schools, which is not unusual for biochemistry. And just to clarify here, it's like a PhD in a medical school, not a medical degree. But nonetheless, because of that, there's always a lot of emphasis on disease and kind of what happens in disease. So this is a little bit of my background, kind of trying to look at biochemistry, but through the lens of how does biochemistry change or go wrong in a disease situation. So the overall topic today is a rare genetic disorder that is present here in Manitoba and the prairies called Bowen-Conradi Syndrome. And it is right now only found in Hutterites. So Hutterites throughout North America have this disease. It's never been found outside of the Hutterite population. So I'm going to start off with a little bit of background, kind of what ribosomes are. You should probably know that because that's really high school biology. So what ribosomes are, why they're important, how ribosomes are assembled. So a different way to say that is how are the protein makers themselves made? And then a little bit about the disease, and then I'll go into some of our data that kind of brings up a new model as to kind of what goes wrong in this disease. So first of all, what's a ribosome? Ribosomes, first of all, are molecular machines. And oddly enough, I've been kind of teaching that on Thursday to my intro biochemistry course. So there's a lot of stuff in biochemistry that is literally a machine. A lot of protein complexes working together. There can be gears, there can be things turning, there can be arms moving around. So it's really kind of almost like engineering at a certain point. And so what you have here on your left is the ribosome. The ribosome has two parts to it. There's the small kind of subunit here in blue, which is what I'm going to talk about. And then there's the large subunit in red. The small subunit, which is also shown here, kind of looks like a duck, right? I don't know if you can see the beak of it right over here of the duck. And by comparison, there is, the projector's not great here. There's actually a small red dot right here. And that's a single carbon, right? So that kind of gives you a sense of how big these things are. So the small subunit actually reads mRNA, reads the genetic code, and the large subunit makes the protein from it. So, oops, okay, here we go. So why is this all important? Well, the ribosome is the one that makes all the protein in the cell. And again, this is kind of high school biology, so you should know what a protein is. But nonetheless, in case you don't, the best example of a protein would be, a piece of steak or, a chicken breast or something like that, right? So it's kind of usually muscle. But it's a lot more than just that. So proteins do things, right? And part of that doing things is things like carrying oxygen from your lung to your little toe. It can also be giving shape to your body. So obviously this is where we go back to muscles, right? Muscles kind of fill out our skeleton and give shape to our body, but muscles, which are made of protein, also allow us to move, right? So that's kind of why I've got, kind of a cartoon flexing here. So a lot of movement is all proteins. And again, that movement is very mechanical in how these proteins work. The ribosome, as I mentioned earlier, is a bit of an intermediate between the genetic code and proteins. So it reads mRNA, so it doesn't read DNA, it reads mRNA, it interprets that mRNA, and it spits out the protein. Just to give you a sense of scale here again, and of importance, in a cell, so heal a cell here as an example, in a cell, there is at least 10 million ribosomes. So from my perspective, most cells are nothing more than bags of ribosomes. However, these ribosomes use up to about 30% of our cellular ATP. So the food that you consume, the energy that you get out of that food, at least 30% of it goes towards the ribosome. And this number also blows my mind. Ribosomes produce approximately 3 × 10 to the 20 new proteins per person per hour, right? So that's just kind of a monstrous amount of work that is very expensive metabolically for the body to do. To really reemphasize the issue of a machine, this is a cartoon that one of my undergrads made for me quite a few years ago. It's a little bit simple-minded, but it will really illustrate the machine concept. So in yellow is the mRNA. Think of, for example, the COVID mRNA vaccine that many people have received. The small subunit is in blue. It's going to do the reading of that information. The large subunit is in red. It's the one that actually makes the proteins from the amino acids. And what you'll see here, if this works for me, pardon me, so there are the 3 sites in the ribosome, which you don't need to worry about, but just watch this go. And it will just keep repeating that, repeating it, till it is actually done making a protein. And then it will go back and start again and make another one. Okay, so this is very much a machine. What it's not showing you is that it ratchets, moves back and forth, and so on. So it really has moving parts. Okay, so why should you care about ribosomes? Well, obviously ribosomes make protein, and our bodies are made up of proteins, so you need those proteins to come from somewhere. So that's a reason. If you're a child and you're growing and kind of growing in height and developing, you're going to need a lot of ribosomes to make a lot of protein because you need those proteins to grow. However, sadly, we've known for about 100 years of this loose association. By loose, I mean not very well characterized, between ribosomes and cancer. And that's just starting to get flushed out as to what the link is. And we can tell for sure that ribosomes that aren't reading right, because remember I told you that ribosomes are reading, right? So ribosomes that are misreading are definitely part of cancer. So I think right now there's a bit of a debate as to whether or not they can trigger cancer or are a consequence of cancer, but I think it's fair to say that they are part of cancer. If you've ever taken an antibiotic, most antibiotics target the ribosome. So tetracycline, for example, as an antibiotic that sometimes children can get, it will target the ribosome. Not ours, but the ribosome of bacteria. and it will basically kill those bacteria by inhibiting their ability to make proteins. And it ends up that a lot of chemotherapeutic drugs that are currently in use, we now realize that those chemotherapeutic drugs block ribosomes, our own ribosomes, our cancerous ribosomes, and people are actually trying to develop new chemotherapeutic drugs that specifically block ribosomes directly instead of indirectly. So the issue here is not necessarily how does the ribosome work. It's more how is the ribosome put together. So a different way of saying this is what makes the protein makers, right? So the protein maker is the ribosome, so what makes it? So the first part of this story is it happens in a specific part of the cell called the nucleolus. And that's something I need you to hold on to, because we'll come back to the nucleolus. So in eukaryotic cells, remember, like big balloon, we have the nucleus where all the DNA is, and then there's a subcompartment of the nucleus called the nucleolus, which is where ribosomes are made. So that's kind of what we see here. This is the nucleus of HeLa cells in green, so that's where all the DNA would be. And these really bright green spots are the nucleoli, and there's more than one, and that's okay. The way that this process happens is almost like a scaffolding. So I kind of think of it almost like, you know, if you're building a skyscraper, right? You've got scaffolding all the way around the skyscraper, and that scaffolding builds the skyscraper from the inside and up. So the ribosome assembly works a little bit that way. So in this cartoon, the ribosome has been turned on its side. So this is the small and the large subunit, but you've got all these workers on the outside that are like working on the scaffolding that is putting this ribosome together. So there's a specific machine that I'm interested in, and this is what it looks like. I really wanted this to be a movie, but apparently movies are not for me today. So this particular thing is called the SSU processome. So we're interested in the SSU, which is the small subunit. So this is the SSU processome. And it's not shown very eloquently here, but in the middle of this SSU, you can see it here in gray, is the RNA that goes into the ribosome. And then on the outside is the scaffolding of all the proteins that kind of really kind of form the assembly steps of the ribosome. We're going to come back to this, but the protein that I'm interested in is right over here in red. This SSU processome was discovered by my postdoc supervisor, Susan Basurga, who visited Yale a few years ago. But ultimately, the work was done by her postdoc, François Dragon, who is now at the University of Quebec in Montreal. And fortunately, I get to hang out with him next week, so I'm looking forward to that. Okay, so this process is important, right? Kind of, we need ribosomes to make proteins to be alive. And it's so important that when I was a grad student, we would often kind of talk in the lab, well, you know, if something went wrong with the ribosome, would you ever know? And we pretty much all agreed that if something went wrong with this, it was so fundamentally important that you would never know that it was going wrong because, for example, a developing fetus would just kind of die. It would stop there. You would never see the final product. And then we were all caught by surprise. So starting in 1999, really in 1998, there was a genetic disease that was found. So Diamond-Blackfan anemia is a genetic disorder. People who have this disease can't make enough red blood cells, so they need transfusions all the time. And it was discovered to be a mutation, a variant, we should say, in one of the proteins that makes up the ribosome. And so then all of a sudden, what we're going to do with that, because it wasn't supposed to happen. And out of that, at about the rate of one a year, a new disease would come out, and a new disease would come out, and a new disease would come out. What we've come to realize is that every time there's one of these diseases, there's not a complete abolishment to the assembly of ribosomes, because if there was, the cell would die. There's more of a slowing down. And as evidence that it can also sometimes go bad, if you have Diamond-Blackfan anemia, sadly, you have a 100% chance of developing a cancer by the age of 30. And the likely culprit is the ribosome making errors in decoding and making errors in reading, maybe putting in the wrong amino acids. Okay, so an example of a ribosome assembly disorder, we call them ribosomopathies, but I think I'm the only person in the world who can actually say that word. So a ribosome assembly disorder here is really Bowen-Conradi syndrome. This is kind of what I think to be a really nice picture. Tim Smith, who is one of the photographers for the Brandon Sun, has done entire exhibits, including exhibits in New York City and New York Times on Hutterite life. And a lot of the pictures that I have are taken from him because they're so absolutely gorgeous. I do have to say, though, that this is not necessarily of Hutterites who have the disease or are carriers, right? This is just Hutterites in the general population, just kind of doing everyday activities. Okay, so our disease, Bowen-Conradi syndrome, some of the carriers here. So 1 in 10 Hutterites will carry the disease gene for Bowen-Conradi syndrome, which is an incredibly high number, right? Usually with some of these rare genetic diseases, you're like 1 in 10,000, 1 in 100,000, but here 1 in 10 is incredibly common. And so that means that 1 out of every 355 live births is a child with Bowen-Conradi syndrome. So right now we have 2 children with this disorder. I'll show you one in a moment. We have 1 child in Manitoba. We have 1 child in Calgary. It's due to this genetic variant. So we tried not to call them mutations, even though I slipped because people don't like to be called mutants. In one of the proteins of the SSU processome. So remember, I showed you the SSU processome over here a second ago. So it's one of the proteins. Let's go back here for a second. So it's this protein over here in red. So it's a genetic variant in that protein. And I'm going to skip over some of the genetics here because it's not all that important. But the children who are born with this disease, they're born very, very small. And then once they're born, they just don't grow. So they stay really tiny. Near my office, I have a picture of a 5-year-old who weighs 15 pounds, right? And so a 5-year-old, I can tell you, having had one, should probably weigh 50 pounds, not 15. And then usually there's death in early childhood. So in the past, these children have died around the age of 13 months. Right now, one of the 2 children is doing relatively well, but mainly because she has a tube that allows her to be fed straight into her stomach, because a lot of these children kind of aspirate their food into their lung, which is obviously not a good thing to do. But regardless, there's not a lot, there's no what we would call curative therapy. So there's no way to cure these children. Right now, we're just kind of keeping them comfortable and, you know, preventing them from choking and so on. So the disease was discovered at the University of Alberta in the late 70s by their namesakes, 2 pediatricians. But if we bring the story back to Manitoba, these are actually 2 of my really close friends, I would say. So this is Dr. Barb Triggs-Rain, whose lab discovered the genetic cause of this disease, the genetic mutation. She's currently a department head of biochemistry at the med school. I desperately want to get her here before she retires any minute now. And on her right is Dr. Cheryl Rockman-Greenberg, who's a pediatrician, but she's a clinical geneticist. So her whole expertise is these rare metabolic diseases. She's lectured in my classes a number of times already. So a lot of my students have actually gotten to meet her, but her whole expertise is really rare genetic diseases in children. And so her role in this project was getting the DNA from the parents and the children and allowing her colleague, Dr. Barb Triggs-Rain, to actually do the molecular work to identify where the genetic variant was. So this is one of the children that we have right now. And I'm going to try to just put out a few like highlight features. So obviously at this point, this child is very, very small because you can see that she's being bathed in the sink. But if you look at her face over here, you can tell that like right where the arrow is, that the bridge of the nose is very, very thick. And you can tell also a little bit over here and over here that there's practically no neck. And if you looked at her sideways, there'd be no chin. So we call these together craniofacial malformations. So the face, when you look at it very carefully, you realize that things are wider than they should, or thinner, or are displaced. And you can see here how small she is. So here she is, she's 20 months old here on the right, but her sibling who does not have the disease, who is only 4 months, is on the left. And you can tell that they're pretty close in size, but they shouldn't be, considering the age difference. And you can also see here, it's hard to see, like the portal where she's fed. And then her feet look like rockers. So like the foot is constantly like this. And also a lot of them will like hold their hands very tightly. And a lot of their knees and their hips are very tight. And that's usually suggestive that neurologically there's a problem there. So as part of my lab, we've been trying to look at like all different aspects of this disease. And so I'm obviously going to present you only one of them, because that's all the time that I really have, looking at the changes in protein-protein interactions. So this is part of what the protein looks like. I might kind of kill the lights here just for a second. So this is part, so the protein comes in 2 parts. So this is 1/2 of our protein. And I'll just kind of turn the lights back on here. So the protein kind of goes like this. So what you're looking at is just the palm up, just 1/2 of it. And what I just want you to see here is this region here. So over here, there's a yellow spot and there's a red spot. So the red spot is the amino acid that is actually catalytic. So it's the one that is doing the work. The yellow spot is holding the red spot in the right orientation. So this is kind of, things have to be aligned just right. So the yellow spot is holding the red spot in the right orientation. And the yellow spot is where the genetic variant is. So right now, I'll show you this in the next slide. There's an interaction, like positive, negative interaction between these two. And in the genetic variant, this is completely lost to an amino acid that has no charge. So that probably means that the other amino acid can just flop around. It doesn't align properly. But in addition, you can see that this yellow spot is at the bottom of a corkscrew area. And that corkscrew area is really important to how these proteins come together. And so we think that with the genetic variant, that this corkscrew area doesn't form, and then the proteins don't come together right. So if we blow this up just a little bit, even though I don't want to spend a huge amount of time on that. So here is our catalytic amino acid, and you can see kind of the work that it's doing here. It doesn't matter what it is, but this is the area in particular that we're interested in. Its main job is to hold this catalytic residue in exactly the right alignment. And in this disease, this amino acid is something else. And that something else, a glycine, doesn't have the ability to have this charge interaction, right? Because this one is positive and this one is negative. So there's a bit of a charge interaction that's happening there that is completely lost. Okay, so one of the main features of biochemistry is that proteins are social. And proteins like to hang out and interact and touch other proteins. They're really social. And people sometimes even draw how proteins interact like a Facebook friends network. And so we know from a lot of other work that's been done on rare genetic inheritable diseases that one of the things that happens in these genetic diseases is that the protein-protein interactions stop happening. So kind of, you can imagine here, if you've got like a circle of friends and they're maybe all linked to that one person, if you remove that person, then the whole circle of friends falls apart, right? Especially if it, maybe it's a family or an extended family or a school group or so on. So knowing that oftentimes genetic diseases result in a loss of protein-protein interaction, one of the first things that I asked in my lab was, well, could Bowen-Conradi be due, at least in part, to a loss of these interactions? So our general theory is this, right? So I'm going to show you the theory and then we'll talk about the data behind it. So what you would have here on the left is what should happen, and what you have here on the right is what we think actually happens. So what I want to show you here is that we have our protein of interest, EMG1. So this is the protein that has the genetic variant in Bowen-Conradi syndrome, but we're showing you here the regular version. And it interacts with another protein called UTP2. Now, in order to make ribosomes, those proteins have to be in the nucleolus, which is a part of the nucleus. So what we think is that EMG1 can't go to the nucleus on its own, but UTP2 goes to the nucleus on its own. So what we think is that EMG1 I might have to go to my computer here. So that EMG1, oh, there it goes, interacts with UTP2. So they have a protein-protein interaction, and together they go into the nucleolus where ribosomes are made and everything is fine. In Bowen-Conradi syndrome, the EMG1 protein has that genetic variant. It's probably misshapen because of that. Remember the corkscrews that I was telling you about? Those corkscrews aren't happening, so the shape of the protein has changed. And then the interaction between our EMG1 and UTP2 is no longer happening. So our UTP2 goes to the nucleolus by itself, and doesn't bring EMG1 along with it. So our EMG1 is stuck in the wrong part of the cell, and it's in the part of the cells where ribosomes aren't made, so it's not contributing to the assembly of ribosomes. So how did we do this? So I'm a big fan of letting the cell do the work for us in terms of the experiment. So we used one of my favorite tests. We call it an assay called the yeast-2 hybrid system. And the yeast-2 hybrid system was invented by this guy over here, Stanley Fields. Stanley Fields retired just a few weeks ago, actually. And he's a big shot. I've had the pleasure of meeting him. And he was at the University of Washington in Seattle. And I think he was a part of the HHMI there and the Fred Hutch there and so on. And his method has been so popular that when I PubMed it, I find at least 17,000 papers that have used this particular method. So this is an incredibly popular method. And just to show you that us yeast people can actually really have fun, you might see, if you've got really good eyes, that the beer that he's holding is labeled Y2H, which is the abbreviation that we use for the yeast-2 hybrid, because obviously beer is made out of yeast. The yeast-2 hybrid uses yeast. So there's a group of postdocs there in Seattle that started their own brewery. And so they made a special brew for him. I asked for it. They won't send it to me, obviously, because it's a different country. Okay, so this is how the yeast-2 hybrid system works. So this, oddly enough, is a hand-drawn cartoon from Stanley Fields' first grant application to get funding for the proof of principle of the yeast-2 hybrid system. And it takes advantage of the fact that transcription factors, which are proteins that kind of decide what genes are made or what genes are expressed, transcription factors are modular. So they almost look like a little barbell or a dumbbell. They have two kind of globular domains. So you can see them right here. So kind of one globular domain here and another one there. And what's really cool about this is that you can cut the protein in two, and it will still work as two independent halves. So what you can then do is attach your proteins of interest to each of the halves. So you can attach your protein of interest to the half that's going to bind to DNA, and that protein becomes the bait. And you can attach your other protein of interest to the prey, and this other half is going to recruit a lot of biochemistry, RNA polymerase II, which is going to come in and make mRNA and so on. And you can attach this in front of a reporter gene, and this reporter gene is going to do something like let the cells live or die on a particular type of media. So oftentimes what we're looking for is cells that are growing or cells that are not growing. So if they're growing, then this interaction happened and something got made here that let them live. If there's no interaction between our two proteins, then this gene is not made. and the cells sit there and they can't grow and they die. So we're looking at growth as a surrogate for whether or not these two proteins are interacting with each other. Okay, so this is work that mainly was done by undergrads in the lab. So this is Courtney Sanjenko, who left the lab quite a few years ago now. She's in her 4th year of dentistry at U of M. This is Trent Nelson, who finished his master's degree in biochemistry at University of Montreal. And here we've got basically 2 proteins. We've got one protein, UTP2, which I've shown you earlier. Right, we've got UTP2, and we've got EMG1 regular, and we've got it in the Bowen-Conradi case. And then just to make it kind of even more interesting, we've got the yeast version of those genes, we've got the human versions of those genes. Even though everything is done in yeast, we still have human genes in there. So we're basically asking, are these proteins interacting with each other or not? And the answer is yes. So UTP2 is interacting with EMG1. So here's a kind of a chart. So here our UTP2 is the bait, and this EMG1 is the prey. So you can see here our UTP2 is interacting with EMG1, so we know that. If we flip things around, they're still working. So EMG1 as bait instead of UTP2 is still interacting with UTP2 as prey now instead of bait. So they work in all directions. However, UTP2 is really no longer interacting with the Bowen-Conradi version. And so here, like the little white spots that you're looking at is actually yeast cells that are growing. And what you need to be doing is comparing how much white stuff you have over here to over here. So you can clearly tell that this would be a lot of protein-protein interaction. This would be very, very little protein-protein interaction. And the same thing down here. So now if the Bowen-Conradi protein is our bait, it now no longer interacts with UTP2, which is there. So essentially what I'm showing through this slide is that when EMG1 wild type and UTP2 are there, they're interacting together. But the minute that the EMG1 in Bowen-Conradi has the mutation, that interaction is lost. So it kind of fits the overall model that I said earlier, that oftentimes genetic diseases are due to a loss of protein-protein interaction. So we wanted to go a little bit further with this. And here, this is a little bit more details, but we kind of played around with quantifying this. So in this particular instance, you can see here with the yeast gene, UTP2, its interaction with the EMG1 wild type is this much. But with the Bowen-Conradi variant, you can see how much less interaction there is. With the human UTP2, it's interacting here with the human EMG1 this much, but with the disease variant that much. So that kind of means that it's true in yeast, it's true in humans, right? Because this would be the human genes in yeast, this would be the yeast genes in yeast. And hopefully I highlighted. We've also decided to have a little bit more fun and say, do the yeast and the human proteins interact? And it's true. So here, this would be the yeast UTP2 interacting with the human wild type or not interacting with the human Bowen-Conradi version. So these are 2 proteins that their interactions are so important that you can actually have a copy of the yeast protein, a copy of the human protein, they'll still interact with each other. If they weren't important, they would not have been maintained between yeast and us. Now, one of the concerns that we always have with genetic diseases like this is, does the variant destabilize the protein? And you would expect that it probably would. And so then it kind of brings up the issue of, if you're seeing less interaction, is it because you've got less protein there? Or is it because the shape of the protein has changed? Or a little bit of both, really. So this is still a work in progress, but this is the work of Danny, who spent the summer in my lab, and I can see that she's there, thank goodness. And just a few things to orient you here. So this is UTP2 here, so that's very large. So anything that is further high up here is larger than, so this would be smaller, this would be larger. So this is our UTP2, which is a fairly large protein. This is EMG1, which is a much smaller protein. And then the darkness of the band tells you how much there is there. And so what this is clearly showing you here as a kind of really nice example, this would be the wild type protein, and this is the Bowen-Conradi variant. And you can tell very nicely from this that there's a lot less of the Bowen-Conradi variant than there is of the wild type. And here, Danny kind of did a blow up on a different gel, and you can really see the difference because they're obviously all the same size protein, but you can tell that on the right, there's a lot less. What we need to finish is like how much a lot less. So at the end of the day, we look at this and we kind of go, well, we're probably losing 90% of the interaction, but we're maybe only losing like 60% of the protein. So chances are that we are really losing more interaction due to the change in shape than to the abundance. So the fact that these proteins are no longer interacting with each other, it's not because you've lost one of the proteins. That protein is still there, but it's changed its shape and it's no longer able to touch its partner. Okay, so obviously we always want to validate these things. I'm not going to go, I was teased a little bit here that I'd be grilled. Usually when it involves a yeast 2 hybrid talk, we automatically know what the grilling questions are going to be. I think I'm safe in this audience though, but with the yeast 2 hybrid, you always want to see some other way that, you know, kind of what you found is not some artifact. And so you shouldn't have to worry about it too much. So these are our 2 proteins. So this is what they actually look like. So in yellow and orange, this is the EMG1 protein that is involved in Bowen-Conradi syndrome. So it kind of works together, as you remember. Don't worry about the red part for right now. And the big blue part is UTP2. So UTP2 is kind of a big like tube, almost like a dumbbell, kind of big balls at the end of it, whereas EMG1 is 2 kind of tight globular domains. And this is not crystallography, this is cryo-EM. So this is from Sebastian Klingel's lab at Columbia, I think it is. And you can clearly see here, again, this would be nicer in a movie, but apparently my technical skills aren't good enough for that. You can clearly see in this area kind of interaction between the blue of UTP2 and the orange and yellow of EMG1. So clearly this interaction is real because we're seeing it in cryo-EM structures. Okay, so one of the questions that we had thinking back about EMG1 hitching a ride out of the cytoplasm into the nucleolus where it's supposed to work is, well, what would happen if we jerry-rigged cells to make a bucket load of UTP2? So this is something that as a biochemist, especially as a yeast person, we get to do all the time. People who do human work have a hard time doing those things because the technology is not there for them. But we can basically like produce buckets. We call it overexpression. And so what Taylor Gooden here, Taylor's now, goodness, I think 2nd year of her master's in public health at UBC. So just to kind of give you a reference point, this is growth. So this is looking at how much growth we have. And this blue line would be how much growth our Bowen-Conradi cells have. And in green would be our wild type. So these are regular cells, wild type cells, and this is the Bowen-Conradi cells. And so you can see that they're growing less well. We figure that's about maybe 60% of wild type. It doesn't look like it's a big difference, but this is a log scale graph, right? So it actually is a really big difference. So when we take these Bowen-Conradi cells and we get them to make a bucket of UTP2, you can see that all of a sudden they grow better. It doesn't cure them, but it at least helps them grow a little bit better. So that is kind of a different way to validate that that interaction is actually real. And it's not really shown here, but underneath this green line would be the wild type cells that are also trying to make a bucket of UTP2, and it's not changing them at all, right? So it's specifically helping the disease version of the protein, not the wild type version of the protein. Okay, so on a parallel project, I had other people looking at the structure of EMG1, And again, so remember it's a dimer, so you're looking at the front face here. The genetic variant is right over here. And so this whole part here, we would call this like a globular domain. It's like a ball, like an misshaped ball. And I started to notice that it had this region here in red, and you can kind of see it go up. And actually, it's been cut off a little bit because it's actually way longer than that. And this region in red, you can see that it doesn't fold up nicely, right? The rest of this folds up nicely. There's these nice corkscrew structures. There's a flat structure in the middle that you don't see all that well. And then there's this kind of random thing. And so we now know that proteins do that. So this is called an intrinsically disordered region. So people get this confused with unfolded or misfolded, which is what happens to an egg white when you cook it. So this is not a misfolded or unfolded part of the protein. This is part of the protein that never really has a real structure. And I think I'm going to say this and I'm going to get killed later on. I think the term that we use in my household, maybe coined by my wife, is the flying spaghetti monster model, right? So it's kind of like these like long arms. And of course, they have the ability to catch things and to grab onto things. So what we have here is the kind of the prediction that that part of the interaction is due to this disordered region here that is very flexible and has the ability to make protein-protein interactions. So if we go back to our cryo-EM structure, the disordered region, which is not all shown because they're difficult to see in cryo-EM, is in red here. And in actuality, it should be much longer, but we see it here better than we see it by other methods. And what's kind of interesting is you can see how much, especially in this region, how much it complements the disordered region in UTP2, because UTP2 is mainly disordered regions. And so we now have this interaction that is between two disordered regions. So one of the things that we're also trying to do in the lab is a yeast-2 hybrid to see how much of these disordered regions can you chop off until you lose the interaction completely. One of the last things that I kind of want to say is, so when I started kind of the projects, there were a few things in the published literature that were pretty much accepted. They were true. Some of them kind of were a little bit difficult to rationalize, but nonetheless, you had to accept them. And one of them was this experiment in the paper that discovered the Bowen-Conradi Syndrome variant. And it's a little bit hard to interpret, but what it's basically saying is over here, if you have one copy that is regular, that is wild type, how much does it interact with the mutant copy? So here, if you've got one copy, so we've got two copies that are regular, so they interact with each other by this amount of strength. And if you then switch it from two wild type to one wild type and one variant, you've got that amount of interaction. So it's interacting stronger. That never really made sense. And then over here, they did it again where they used the variants. And again, they did, I think, one copy that is bad, one copy that is good, and then both copies that are bad. And they got even more interaction. And that never really made sense to me, mainly because the fear there was that you could get clumping instead of protein-protein interactions. Because when you cook your egg and it changes kind of translucency in your pan, that's because the proteins are clumping as they cook and unfold. So that was a bit of a problem because our data was the opposite of this. So our data contradicted that data. And at the end of the day, I'm going to spare you some of the details, but when we do yeast-2 hybrids, we have to put in an inhibitor to kind of tamp things down. And when we put in the inhibitor, as we're supposed to do, the mutant protein no longer really interacts. So it kind of behaves properly. But when we don't put in the inhibitor, then everything starts to stick. So now we've got a bit of a problem because we're trying to change what the literature is saying, but we're also able to kind of point to the other paper as to what they forgot to do. But now we have to probably back this up with a whole lot of other experiments to really be convincing that we're right in that particular So what you're seeing here is this would be the interaction under normal situations, but with the disease situation, these proteins are unfolded. That's kind of why they're shaking and they lose the interaction. But that is because we have the inhibitor present. When the inhibitor is absent, then we actually see an interaction, which we don't think is real. Okay, so that I think is in a nutshell the talk. Just to point out that we are obviously working on a lot of other aspects of this disease, one of which includes, I mentioned earlier in the cancer situation, the ribosomes making errors. So we don't know if they're making errors, but we do know that the ribosomes in those kids aren't working as well. So we know that there's changes there. And we're also looking at things like, well, what could we do? Are there any drugs that we could be giving to these children to help them out? It may never cure them, but it might help them a little bit. So my kind of research program around this particular disease has a lot of different pots, and we've got our finger in pretty much every single pot, which is probably we're a little bit overcommitted. I'm going to stop there. So this is a list of pretty much everyone who's been in my lab, highlighting the people in red who've contributed in some way to this project. I also want to thank some of my collaborators, but especially Dr. Cheryl Rockman-Greenberg, who's been a real support in actually kind of getting a lot of the work done on this particular disease. I thankfully get funding from NSERC and CFI, but I've also received funding from the Children's Hospital for this project, which I'm very, very thankful for. And I'm going to leave you with an old paper, an old photo of the lab that desperately needs to be updated. Taylor is here, who's did the overexpression with UTP2. Courtney Gear repeated the yeast-2 hybrid. So it was done by Courtney Senjenko, but Courtney Gear repeated it to make sure that we were on the right track. And some of the other people were working on slightly different projects at the time. So I will end there. I'm not over time yet, and hopefully I can take some questions.
Go ahead. Okay. So you were saying, just to confirm, that there is a loss in the protein abundance of EMG1, and then you were saying, so you're not sure what the ratio of loss in protein interaction and then protein abundance is. Is there a certain way you would go about determining that, like as a next step? Or is that something?
So right now, my knee jerk is that there's more loss of interaction than there is loss of abundance, even though the loss of abundance is still significant.
Okay.
So what some of the previous students have been able to do is to quantify how much interaction there is. So we can at least get the numbers on that. When it comes to quantifying the protein, that's where it becomes much more difficult. So this is kind of really the work that Danny spent over the summer doing, I don't know how many Western blots in a day sometimes, to kind of make sure that we're putting equivalent amounts of protein in every lane, and then quantifying how much of the EMG1 is within that lane. And one of the places where it gets a little bit tricky is that EMG1 influences how much protein is made by the cell. So then you have this slightly circular argument, and you have to kind of like try to rig it right so that you're looking at how much protein there is, not the circular argument of, well, there's less of this protein, so therefore there's going to be fewer ribosomes made, so therefore there's going to be less protein translation happening. So it's a little bit trickier.
Okay.
Yeah, but it would be just kind of then doing quantification. I think right now we're at a point where we would just need to quantify how much protein is in the lane, how much EMG1 protein is there. We've already got it done in triplicates, and just kind of do the math and get the number.
Okay, thank you.
I have to do a lot of reading on this, but my gut for that type of situation where you have possibly causal or explicitly circular causal situations, it's floating. It's in my list. It's statistical causal analysis can take quantified numbers like that in a situational tree, and then you can use the variances to make the determinist instead of using statistics inferentially, you can use them to extract the logical path of causality. I'll send you an.
That's way beyond my
No,
statistics
no, no.
skills.
You'll take a set of 8 states, right? The situations that you have from your experiment, and you can unwind the circular causality that breaks your argument, right?
I can see where you're going.
Okay.
We would need to have our hand held, but I can still see where you're going.
I'll get there in the next year.
Sure.
And then we'll okay.
Yeah. So Mike, the thing you did not discuss, which is that the cancer part, but I'm not going even in detail there because there is already, you observe a reduction in overall production of ribosome, right?
Yeah.
So I'm just putting that on the disease model and thinking that, are we talking here like the ribosome, or we are talking about the processome, or we are talking about, do we have some knowledge from literature already what exactly happens? Because if there is a problem with the ribosome making overall, then it's kind of everything affected, right? So sometimes it is like, if you are expressed everywhere, sorry, I cannot make you a target. You understand like what I'm trying to go. So if it is overall production of the ribosome, that's a bigger problem than it is just the assembly problem, or it is about the transportation as you have shown us. So what do you think exactly the problem could be if there is a reduction? Is that because they're not assembling well, or there is some way the production of ribosome is inhibited?
So I guess Dr. Majanda's question is really asking me about kind of what's the ribosome link to cancer and to these diseases. And she's already hit kind of a number of problems that this field is struggling with. One is that in a lot of cases, there's less ribosomes because of the genetic disease, but then in cancer, there'd be more ribosomes. So how do you justify that? And usually it's kind of the justification is that you'd probably get multiple mutations. So the first mutation would probably decrease how much ribosomes that you make, but then a second mutation would probably select a population of cells that have ribosomes that are maybe sloppy and allow that population to then expand. I think it's all kind of a lot of arm waving at this point. There are assays that we can do to look at how accurate the ribosomes are. Right now, our assay only shows us that there's less protein production. It doesn't show us as to how accurate the production is. But in this cartoon here, there's 3 sites, and this P site is really important. And it has there's a uridine base that has all these decorations on it. And so EMG1 is part of, I skipped over this because it was going to be too much. It adds a little methyl group to the P site. Now, the issue is that there's a paper out of UBC about 2 years ago that shows that 50% of colorectal cancer cases, the P site is not properly formed. So then you have, again, a bit of chicken and egg question, is it that the cancer caused the P site to be misassembled, or is it the P site was misassembled to begin with and it caused the cancer? But nonetheless, the fact that it is, partially missing in 50% of colorectal cancers tells you that something is going on there.
Thank you.
I think you just missed the point. What was the etiology behind this loss of PPI in this syndrome? Was it because of the wrong interpretation or reading of the mRNAs, or the amino acid sequencing, sequencing wrong, or both of them?
So it would start. Well, maybe this one is easier to look at. So it would start with a genetic variant where this is no longer an aspartic acid, it's a glycine. So that would be in their genome as a glycine instead of an aspartic acid. And then it doesn't hold this one correctly, and this doesn't get, this is the uridine I was talking about, doesn't get this done right. The theory that is here is that once this isn't done right, then that site now no longer properly properly reads the mRNA and could then cause additional mutations, variants, but at the protein level, not at the DNA level. And of course, those protein variants could then feed back at the DNA level. So then it becomes really quite circular. But the location where the new mutation comes in is protein, not DNA, which is in itself novel. No, so we don't know if they're being affected, the disordered region. It was more the surprise that they were there.
Oh, okay.
Right, and that they seem to be participating in the protein-protein interaction.
Okay, so the variant doesn't cause anything on the disordered regions? Or, sorry, did I misunderstand? The disordered regions are only in the EMG1 variant protein?
No, so the disordered regions are everywhere. But where it gets messy is that the disordered regions are really close to where the variant is. Right? So like the variant is right here. So it's, I can't remember, it's either the first or the second amino acid of the alpha helix. So it's clearly in a structured region. But there's a disordered region here that abuts it. And there's another disordered region here that is a little bit before it. So I don't really know what the role would there be, because I would think that if this is an aspartic acid to a glycine, and we have good reason to think that you probably lose that alpha helix, then you could probably extend the disordered region, but then it would be unfolded, not disordered, which is not quite the same thing.
Okay, thank you. Just interested.